talk about the item 6, which is about white matter imaging. And in the white matter imaging, we are talking about what we call diffusion tensor imaging, white matter tracks, and how we can detect signal within the white matters to be able to show those white matter tracks. And we discussed about the white matter tractography before in the previous sessions. But today, just giving you a sense about the type of signal that we are talking when we have water molecules in the especially in the axons. They have a specific direction, and then when there is a specific MRI signal that is measuring the level of the direction of the water infusion. And that specific signal could be detected from MRI. So we can see what are the areas that have a specific direction of water flow. And based on that, we can say that, okay, it seems that there is a pathway there that is kind of transferring water alongside, and that is going to be a way to be able to detect those white matter tracks inside the brain. And obviously, when we are in the areas like cortex, the signal is just homogeneous. Water would be distributed equally in different directions. But when we are in areas that are related to white matter tracks, there is always a direction, because these white matters are giving directions. But these are not directional. So there is no single direction. And based on that, we can make these beautiful figures. But the challenge is about the areas that we have one fiber crossing the other fiber. So when you go to these areas, there are some signals in one direction, some signals from the other direction, and you cannot differentiate them from each other. So what you will detect in DTI, these areas that we have cross between different fibers, these areas are challenging to detect. And so what you have these directions, these beautiful directions still here, you have these beautiful directions still here, these are directional, so you can make these tracks, but here there is no direction, so you will lose how these tracks are connected with each other. So if you use these traditional ways of measuring DTI, what you can here in corpus callosum, you can see here, these are well connected to each other, these areas. But when you get to the area that they are crossing with each other, you cannot follow the fibers here. Now there are new methods for DTI that we can detect how different fibers are crossing each other, and then how they are pathing each other towards other directions. So with the new DTI measures, we basically solve this problem, and now we can even have higher quality DTI measures. And there are different methods for that, for solving the cross road problem in DTI. One of these methods is called kind of DSI or cue ball sampling. Some of them are other methods like ST, spherical similarity methods. There are variations, something like 20 different methods to solve the problems, this specific problem. And I'm not sure that it's going to be really helpful for you to know about the details about them. I think they're basically too much for people to learn. And there are some other methods like the HOMA method that we have, or HMOA method that we have here, that could give us better quality DTI. I mean, these are the minor technical details about DTI. And probably, for example, in our center that we are doing the imaging all the time, we have 2 MRI scanners in our center. From something like 40 researchers that we have in our center, probably one of them is kind of understanding the technical details of different methods. We just refer to that scientist who is doing the kind of specific DTI measurements. But this is not something that you use in a kind of regular way. But you know that there are different methods to just improve the quality of these tracks. And at the end of the day, you can have these beautiful white matter tractography. And over time, you start to realize how these things are connected to each other. Especially when you see them in something like 3-dimensional map, they are just really complex to differentiate. For example, last session we discussed about, if you remember, we discussed about arcuate fasciculus. So if you want to find arcuate fasciculus, which one is arcuate fasciculus here? So in the three-dimensional way, it's just getting too complex. Here, it divided arcuate fasciculus in three parts, as you can see here. Arcuate fasciculus, this is the long part, and then this part, and this part. So they kind of divided arcuate fasciculus to three parts. And then what here we call the uncinate fasciculus. So it's always important for you to start to learn them gradually over time about these white matter tracts. For example, let me just give you an idea about this tract, frontal Aslan tract. This one is an area, if I just make a section, this one is a coronal section from the frontal part, and this one is superior frontal gyrus, and this one is inferior frontal gyrus. This specific bluish tract would connect these two parts from each other. And why it is important? These are basically some inhibitory control areas. So if this specific tract is disrupted, you would lose a level of self-control that you have. So you need to start to learn about these tracts one by one in a two-dimensional way first, and then giving them a chance in the three-dimensional way. Okay. And then the last slide in this part is about how we basically combine these pieces of information from each other. So how we combine the information that we have from the EEG, from fMRI, from DTI, to make a map from different parts of the brain that are interacting with each other. Okay? And that is the beauty of the field. So what we can do here, for example, here, you can see that, for example, medial or posterior-medial frontal cortex is oscillating in the theta rate, interacting with dorsolateral, this part of lateral posterior part of the frontal, and then the visual cortex. This is when you are in a high level of attention. When your attention is high, these areas are in theta and gamma activity, which means they are, theta is for connection, gamma is for high processing, and then these areas are silent because these are basically default mode network which are related to self-referential processing. When you are attending to the outside environment, so you are going to do a specific, let's say, exam or something that is outside you, you need that level of attention, and you need to have high level of executive functioning, and also you need to have your vision really activated, seeing what is happening outside. You probably are not interested very much to kind of listen to a music or have information from the auditory part. So you can turn on or turn off different parts with these connections, with these what we discussed in terms of the white matter and different DTI measures and EEG, and how these things are going to get connected to each other. And the synchrony between these areas, as you can see here, are being processed by theta synchronization. Okay, so these areas are, when the areas are supposed to get connected, we have theta phasic synchronization, or we have sometimes areas that are connected with low frequency fluctuation. So these fluctuations are going to be important. And then from this point, we will start to think about, okay, how can I use this information and do the stimulation in the brain?